International Conference of Biodiversity 2023. Before we delve into the heart of our discussions, I would like to express my great appreciation for your presence here today. And please allow me to extend my heartfelt gratitude to Her Royal Highness Princess Mahachakri Sirinthorn for gracing us with her inaugural address this morning. Her dedication to environmental conservation has been an inspiration to us all. The morning session has been nothing short of extraordinary thanks to the insightful contributions of our distinguished keynote speakers. They have shared their experience on critical aspects of biodiversity that resonate with the theme of our conference, Biodiversity, Key to Better Life. Our esteemed speakers have offered us a broader perspective on biodiversity. Their talks have set the stage for the diverse discussions that will unfold over this afternoon and tomorrow. As we transition into the heart of our conference, it's time to delve into our first session, focusing on biodiversity and ecosystem conservation and services. This session brings together the expertise of 3 invited speakers and features 5 oral presentations, all shedding light on the critical intersection between biodiversity and the conservation of ecosystem and services. Allow me to introduce the distinguished professors who will guide us through this session. Our session chair, Professor Dr. Savitri Limthong, the Director of the Biodiversity Center at Kasetsartศาสตร์ University. And Professor Dr. Somsak Panha, the Director of the Center of Excellence on Biodiversity at Chulalongkorn University. Now, without further ado, please welcome our chairman, reporter, speakers and presenters on the topic of biodiversity and ecosystem conservation and services.
Thank you for introductions. So, should I say, good afternoon, ladies and gentlemen. It is a great honor to welcome all of you to our first session in biodiversity and ecosystem conservation and service. As introduced by our MC, that we will have 3 invited speakers and 5 oral presentations. So please join me to welcome our first speaker, Professor Robert G. Coles. He is from James Cook University. His title of talk is Managing and Protecting Seagrass Resources in the Topics in the Face of Climate Change Lessons from Research in Northern Australia. Professor Coles, please. Yeah.
Thank you, Madam Chair. And very much appreciated the opportunity to come back to Thailand. So thank you very much to the organisers of this workshop. It's really great to be back in Thailand. I've travelled here before. But today we're going to the marine world just for this talk, away from the tropics, away from the rainforest, into the sea. We have a research centre at James Cook University. We've been active since 1981, so we've been there a long time. That's why my hair's gone grey, because I was there in 1981. And we've been working on fisheries and seagrass since that time. So I'm going to take you through a little bit of our work today. So this is what I will do. I'll talk a little bit about where we're working, because that's important. I'll give you a very quick lesson in tropical seagrass biology, because I come from a university and I can't miss that opportunity to teach you. Apologies if you already know about seagrass. I'll go back to our research and monitoring history, the importance of data, because that's very important to understand in a changing world how important data is. A bit about connectivity recovery, and then if I have time, some climate change. So if you think of going to Australia, of going to Sydney Harbour and looking at the Opera House, that is not where we work. We work up in the far north. You can see the picture over on the right-hand side there. We're based in Cairns, which is about as far south of the equator as we are north today. And we work along about 6,000 kilometres of coastline from Gladstone round to Nhulunbuy. They look like towns on the map, but they're really quite very small places. This is a world where it is quite difficult to work because there's not a lot of backup. When Halpern did his global assessment of the places in the world which were still in good condition, these are some of the places that he mentioned, not because we're good managers, because they're just very difficult to get to. So look at the helicopter picture there on the right-hand side. That's near Karumba. I can show you where that is, down here. You can see the wetland there. This is one of the amazing wetlands you get in this part of the world. They're inaccessible for most of the year. You can drive. That's a picture of my car out on a road out in the desert, but if it rains, you're going to stay there through from now till after Easter, after April next year. So this is a place that's quite difficult to work. There's a lot of coastline. There's not many of us. And you'll see that when we have data, how much emphasis we put on making the best use of that data because it's so very hard to collect. So then a quick lesson in seagrass biology. Seagrasses are true flowering plants, angiosperms. When they live in the water, they can complete their life cycle completely underwater. They have a true root system, they have a vascular system, so they're not algae, and they're more related to lilies and gingers. They're not really grasses at all, although we use that term. They need light. They're a green plant, so a key essential is they need light. If you make the water dirty and no light gets to the bottom, you won't have a seagrass meadow, it'll die straight away. And they have a general structure. I've just put a couple of pictures up there of a horizontal rhizome with roots coming down and leaves coming up. But the seagrasses themselves come from different lineages. So if you're a dugong and you eat one of those strap species off, the plant can grow new leaves. If you do it on the round leaf ones down the bottom there, that plant can't regrow those leaves. It has to put out a runner and new petioles and new leaves. So they're quite different in terms of how they survive in the wild. When people talk about seagrass, you're putting together a whole lot of different plants that may act quite in different ways. But they all need light. I've got a couple of pictures down the bottom. Some of our seagrasses are very sparse. Some can be a metre tall. We share about 10 species with Thailand in Northern Australia. We have some you don't have, you have some we don't have. But they're flowering plants and you can find the flowers. So down in the middle, the picture there, you can just see the very fine female flower on Thalassia, on Halophila spinulosa, sorry. These are very small flowers. They're only a couple of centimetres big. There are no hibiscus flowers under the water. There are no big, pretty flowers to attract bees. But they're still very beautiful. And once you've found one, you can find them quite easily. You just have to sit in the mud with a glass and look very carefully, and you can find them. To me, they're very beautiful. I managed to take 7 different seagrass flowers to a wedding once, and it was just not appreciated. It smelt and it was a bit wet. And they didn't understand how good that was, getting 7 flowers at one meeting. But over on the right-hand side then, Thalassia flowers, and you can see the seeds. They're quite easy to find. Of those particular plants, you just have to sit in the mud and sieve the mud and you'll find the seeds. So these are proper flowering plants. One of them has to come to the surface. You have that one here, and you can see the big flower on the top left-hand side there, Inhalosaccharoides, has to come to the surface, and it will match with male pollen, which has sacs, and you can see them in the water there. Some people think that's polystyrene foam when they see it, because every month you'll get that white on the surface, which is actually just a seagrass flower. And they're important. And they're not wilderness plants. The top picture there is in one of our biggest ports in Queensland, a big coal port, Gladstone. Plenty of seagrass there. But what they are is they're the primary producer in the bottom of the ocean. So they're growing all the time. They're absorbing carbon and nutrients from the water. They're feeding fish. They're providing shelter. These are very important parts of the marine environment. And it's very important we look after them. They have very high leaf turnovers. So anything that you see green there now, in 10 to 15 days will have gone completely, grown off, broken off, been eaten, or become part of the refractory carbon in the ocean floor. They are in flux all the time. It's not like a coral reef, it's always there. They are growing, being eaten, breaking off. And in doing that, they absorb nutrients and chemicals from the water and effectively act as a giant aquarium filter that you don't have to pay for, keeping the water clean for corals and other things. And they do store carbon. And I've just got a picture down the bottom. We do quite a lot of work putting cameras out, watching what's in the seagrass, counting the fish to estimate for our government how important the seagrass is. And you can see how important they are for biodiversity. Lots of things living in the seagrass. We have a big prawn shrimp fishery. You can see the prawns and shrimps there. And the grey tattler there is part of the East Asia flyway, which is so important for the bird people. The birds that fly from China and Siberia down to Australia and back stop at meadows in Northern Australia and in Thailand. So very, very important in terms of improving the biodiversity in the bottom by providing a structural component in what would otherwise be a flat bottom. Really important. The crocodiles are also important too. In the picture over there, we have lots of crocodiles, which sometimes affects how we work. And I just wanted to show you how much productivity you can get from a seagrass meadow. So the top picture up there is me standing on a seagrass meadow in southern Australia. This is a place where you get the big Posidonia meadows. And after a storm, there's a pile of carbon on the beach there about 3 metres deep, and you can see where it's been put up there before. These are amazingly productive plants in the temperate areas where you can get tons and tons of plant material on the shore, which is actually taking carbon out of the ocean. You don't get that in the tropics. On the right-hand side there, I've got the best I could find in the tropics, and they did not last long. That was eaten very quickly. But in the tropics, you do get these species that aren't very tall. The actual green bit's quite small. But if you dig a hole, you can see the hole there, you can go down a couple of metres, and the seagrass roots go right down into the bottom, and the plants are absorbing oxygen and pushing that down to the roots and creating an oxic layer, a microbiome in the ground, which is very, very important for small creatures, for bacteria, for small shells, for worms. So there's an entire biome underneath the seagrass meadow, which is really important for biodiversity. You can see that too on the right-hand side there where I've got the arrow. That seagrass meadow is holding that sediment in place so it doesn't remobilise and doesn't cause the water to become dirty. So really, really important, and something that is not seen normally. You don't normally go out and dig a hole in the mud flat to see what's under the seagrass meadow. And it's important for grazing, and we had a student who's now has a doctorate, Abby Scott. She put out cages on the seagrass meadows at Gladstone in this case, and on the top left-hand side there was a fish cage to keep small animals from feeding on the seagrass, and after a month or two you can see the growth inside the cage was much better. Bigger cages to keep turtle and dugong out also showed an increase in the actual seagrass in the cages. So the seagrasses are being fed on all the time, and people come to me and say, oh, the seagrass is not looking particularly good, it's very short. And the answer is, well, we may have done something wrong, or it may just be being fed on in the normal process of ecology. And we know Abby put cages out at a reef site near us. You can see there the August picture, the cage was there and it sat there like that for several months. And then in October, we came back and it had been grazed outside the cage. And we know who the culprit was because we have cameras and we can watch. Is that going to come up? There should be oh dear, there should be a turtle turn up there. I'm not sure how I can start the video up. It's not going to work. So in your mind, you have to imagine a turtle swims around and looks inside that cage. The video isn't going to work on this screen. So I'll drop back then to our research monitoring history. We started back in 1981, mapping the seagrass in Northern Australia. I have to explain to you what it was like in 1981. There was no mobile phones, there were no laptops, there was no GPS positioning fixes, machines. You want to know where you were, you used a radar or a compass or a sextant. And we made maps. There's our first publication by sticking little stuff on the map called Letraset and taking a photo of it. I'm sure very few of you will remember back to the 1980s and how primitive it was. If you wanted to do an analysis of variance, you did it with a pencil and a slide rule. How things have changed, and that's one of the things I'd like to point out in this talk, is that even though we're faced with climate change and we're faced with lots of threats, our ability to understand these systems and to measure and monitor them has increased at a much greater rate than the change in the climate. And since the 80s, we've monitored every port in Queensland every year, and we've done a variety of mapping exercises. So if you come down to the bottom then, you'll see that we've got 129,000 records. Now, we have many more data points than that. 129,000 records is the ones where someone has gone down and touched the seagrass, so we absolutely know it's seagrass. It's not pictures from an aeroplane, it's not drone pictures, it's actual collections that we've made. And we've done that using lots of methods. You can see down the bottom, we've crawled on the mud, we've used motorbikes, we unsuccessfully. We had a dangerous hovercraft for a while. Mostly these days we use cameras because of crocodiles, we can't swim very easily. And we do a lot of work from helicopters. Now that's something that's particularly for Northern Australia. We have lots of helicopters because they use them for mustering cattle. They're cheap to run and our labour costs are high, so we can work very quickly from a helicopter. You can see the team there just dropping a quadrant out the helicopter door and actually doing an assessment. And that's me at the very top with slightly less grey hair. Back in 1980, collecting prawns from a seagrass meadow. What we've done with that data, though, is we've gone back in recent times and we still had the old paper copies from 1980, and the three of us still from that original team have gone back through that data and checked it for position fixing, checked it for taxonomy, because taxonomy's changed, and re-entered it into a digital database so it's available. And we've made that available, and you can see the dots there. You can see the 6,000 kilometres of coastline. The green dots are seagrass. The grey dots are where we didn't find seagrass. And very fortunately, we kept both sets of data where we didn't find seagrass as well as where we did. And we have put that up on a web portal, and that's available under a Creative Commons 3 licence for anybody to use. You can use it in three ways. You can read our reports, which is a simple way. You can use a simple online sorting device online. You can look at times and species, or you can download shape files, and if you have that expertise, use it in an ArcGIS or whatever GIS form you use. So a very powerful tool. We've made sure that data now is safe, so it can't be lost. It was on paper form. It's not going to be that safe. It's safe now. And you can go in there and you can click on a dot, and it'll take you back to the original data, that case in 1996, and you can see who collected it, where it was collected, and what was found. So a very, very valuable data set that we have really, really decided to use in extensive ways. So the first way is just in a straight PR way to say, look, when you live in Queensland where I live, all you hear about is the Great Barrier Reef and the coral, which is really, really important for biodiversity. But that reef encloses a massive lagoon, a fabulous lagoon full of whales and dolphins and sharks and rays and crabs and prawns. A very, very diverse lagoon which doesn't get nearly the publicity that it should. So you'll see up there, I'll go to talks and say, yeah, Great Barrier Reef's great, but there's actually more seagrass in the Great Barrier Reef than there is coral, and you need to know that. It's one of the largest ecosystems. We have nearly 20% of the world's species. It's intertidal down to 76 metres deep, which is important to know. The water is very clear. Plants can live down that deep. The light gets down there. And it may not be quite what you think it is in terms of density. I've put two figures there, 35 to 94,000 square kilometres. The reason they're in red is because nobody can map a 300,000 square kilometre marine park down to 76 metres. The only way you can do it is some form of modelling. And how you model it and what parameters you put in can give you a variety of answers. I've given you the outside range there. Think, though, that it is a large area. You don't really have to come up with the figure. It's also worth noting, if you look at the pictures down the bottom, that a lot of that area of seagrass is not your classic meadow. It's actually very, very sparse, halophilous, and many of those species are annuals. They're not there through winter. They're only there in summer. They'll seed, the seeds drop into the sediment, and they'll regrow again come spring. So they're not always there. But you can get quite dense seagrass meadows. The picture on the right-hand side there is a reef top near where I live, Green Island. I really recommend you come to Cairns and go to Green Island at low tide, because you can find 9 species there just in a casual walk on that reef top. People come to Green Island to look at coral, but it's actually one of the best places in the world to look at seagrass. I recommend it. So how we've used that data as well then is to provide advice to management. I've just brought out a couple of cases here. So early on we did a Bayesian modelling exercise just looking at the probability of any seagrass being present. You can see on the left-hand side graph there, the green is just a straight probability of seagrass being present using a Bayesian model from what we know where seagrass is as a parent model to estimate where the seagrass is. And you can see it actually shows quite big subsections in the reef. Around where we live, it's dark green, lots of seagrass. Further down, we have really big tides, 8-metre tides, and seagrass doesn't like bottom stress, and there's not a lot of seagrass there. You'll see it's going yellow. But we use that initially. We have a trawl fishery, and you can see from those pictures that trawling does disturb the bottom. And we were asked to say whether trawling should be stopped in the Great Barrier Reef because it may be killing seagrass. But the trawlers in our part of the world all have transponders on board, and we can interrogate that transponder to see when the boat was going slowly and trawling. And we're able to overlay, and the second picture there is us overlaying the trawl pattern over the seagrass. And I'm not going to show the graph, but the answer was about 20% of the catch comes from grids with seagrass, but they're only trawling about 5% of the reef bottom more than once a year, and one trawl pass is unlikely to make a lot of difference. So we were able to say to the management authority, look, trawling's not really a problem. We do have problems, but trawling's something you can probably put to one side. So incredibly valuable data when you can extract straight into management to make decisions, a really valuable exercise. But in more recent times, and I'm very briefly going to say this, we have enormous amounts of environmental data if you work in the Great Barrier Reef. We could find 13 data sets that are available, things like bottom sediment, lights, high light, low light, currents, wind shear, all those things are there in digital form which we can use. And in the last few years, last 10 years, we've managed to get these incredibly good machine learning statistical processes, these ensemble statistics, if you like, regression trees and random forests, and we're able to put our knowledge of where seagrass is and where it's not into that model and train it. You use some of your data to train the model, and then you can look and see where that model will predict that seagrass to be. These are techniques we did not have a few years ago. The statistical world, modelling world has really, really moved ahead. So we use that seagrass data, environmental data, and I've just got one output from it. And you can see we can now have what we call a habitat suitability model. This is not where you find seagrass. This is where the habitat is suitable for seagrass. And if you're talking to a management agency, you would say, look, even if there's no seagrass there, this is something perhaps you should be looking at protecting because it's somewhere where seagrass should be, because we have a model that has been trained on where we know seagrass occurs, and we can then spread that out across the whole reef. And you see the colours, the different probabilities, and that's one of the problems with modelling is that you can change the answer by changing the probability, and that comes down to more expertise than it does science. What it did show, though, was almost all our seagrass is one genus, Holophila. So the ones you can actually see there at that scale are the very ephemeral colonising Holophilas. When you get to the structural ones that people talk about, the shelter prawns, they're so small in nest distribution you can't even see them at that scale, and I've blown them up for you here. Zostra down the bottom. They only live on a very narrow strip of the coastline, which is important to know because if people start developing that coastline, it's those species that will come under threat, and it's that biodiversity that will be lost. This is not published yet. Wait till next year, we'll finish it off. But it's becoming a really, really interesting exercise in developing these statistical modelling processes. I will step back now, though, to what happens when it all goes wrong. Very topical at the moment because there's a cyclone about to hit Cairns. In 2011, we had Cyclone Yassi. The 294-kilometre wind recording came from about 20 kilometres from my house. So I can tell you what it's like when the wind blows at 294 kilometres an hour. It looks like the picture down the bottom. It's devastating. But even more devastating for seagrass. We couldn't get in the air to map the seagrass and check for about 5 weeks after the cyclone. Even then, you can see from the picture, the weather was awful. It just rained and blew. But more importantly, if you look at the watercolour, it's after the cyclone, it flooded and we got all that turbidity inshore, so no light was getting to the bottom. And even more, there's all sorts of debris in the water, old logs that have floated down rivers, washing backwards and forwards, a totally toxic environment for seagrass, and it completely wiped out the seagrass in that area. We know how it went, though, because just north of there we have a monitoring site we monitor every year in Cairns Harbour, and we track the changes. If you go to the left-hand side there, green is good, red is bad. The dotted line is the 10-year average, and you can see the area of seagrass in Cairns Harbour started going down. We had Cyclone Larry and then Cyclone Yassi. By the end of that, there was no seagrass in Cairns Harbour, and you can see the pictures before the cyclone, then there's bare mud, and then it came back from deeper water, moving into the shallows. We also trawl in that seagrass meadow, so we know what fish and prawns are there, shrimps, and you can see pictorially there. When the seagrass there, we had massive biodiversity. We had 20 species of prawns, about 100 species of fish. When the seagrass had gone, it was down to 1 and 2. We are going out again in February if this cyclone doesn't knock it all over again, and we'll have a post-cyclone measure as well. This is all published, and you can look it up in scientific reports if you want to. But the seagrass was really key to the biodiversity in Cairns Harbour, but it also note that it took nearly 3 years to come back, and if you're a turtle or a dugong that needs to feed on seagrass, 3 years is a very long time. We think it came mostly back from seed banks, from seeds in the mud in Cairns Harbour, but there was also must have been other ways of doing it, and that's led us into a different process. We had a student who's now Dr. Samantha Toll. Samantha said, look, it must be like the rainforest, where birds move seeds around and they defecate and the seeds grow, and so birds are very important to move seeds around in the rainforest. Why wouldn't you expect it to be like that in the sea with turtles and dugongs? So she took me out and made me follow turtles and dugongs around for weeks with a net and catch their feces when they float to the surface. And you'll see they're holding a dugong poo. It is as unpleasant as it looks. It smells. It's awful. I don't recommend it at all. But she was adamant that we would find seeds in it, and she took those feces back to the lab and put them under the microscope. She had the lab to herself because it smells. And she found, yes, there's around about 3 seeds per gram dry weight of feces. I don't know what dry weight of feces look like, but and nearly 10% of those seeds were viable, staying viable, and we've since grown some of them. We found 4 species, but the thing about turtles and dugongs, they have a very long gut retention time, around about 100 to 300 hours. So in that time, they can move somewhere between 200 and 600 kilometres. And there's lots of turtles and quite a few dugongs. So on any night of the year, Samantha would say, there's hundreds of thousands of potentially viable seeds being shifted around. And the advantage of using dugongs and turtles to move seeds around is they're almost certainly going to go to places where they would expect there to be seagrass. So they'll defecate with those seeds in the right place. The other thing I'm not showing today is that Sam also did germination studies to show that seeds that have been through an animal actually germinated better. The passage through the gut has some scarifying effect and removes some of the fungus and diseases that seeds get. So really interesting. The other thing we did, and with her again and another student, is we noticed that if you go around in the water with a net, you'll find anywhere in that shallow water, you'll find viable fragments of seagrass. These are pieces of seagrass with a growing tip, with a meristem, that if they landed on the bottom could grow. Now, Samantha found about 0.6 viable fragments of seagrass per 100 square metres of water, which doesn't sound much, but in actual fact, there's a lot of water out there, so it's quite a lot. So we thought, well, maybe that's another way that seagrasses come back after a cyclone. So Alana Gretchen and I then used a model. Now, I'm not going to go into the details of the model. It's a slim model. It's a depth integrated open grid model of where water flows. So it integrates wind speed, water flows, tidal flows, and it shows you where the water moves. So my videos, is my video working there? It is working this time. It shows you where the water moves in any particular place. And we use Townsville, just south of Cairns, as a focal point, and we used 33 meadows we'd mapped, and we ran a simulation with 68 simulations, 34 with things floating on the surface where they were wind affected, 34 in the water column. 154,000 seagrass particles went out in each simulation. We used real time, so letting them go at different tides, using real wind in November 2011. A decay rate, a binomial decay rate to allow for things to die, and an 8-week maximum time. And we set these propagules out seagrass propagules out. Now, the actual no seagrass were hurt in this process. It's completely a simulation. It's just Lagrange particles, but they reflect what a propagule of seagrass could do. Mostly it told us what we thought would happen, and that is that the average distance that propagules would move was around about 40 kilometres, 50 kilometres. What did surprise us, though, was the outliers in that box plot, and they're as long as 900 kilometres. So what it meant was that propagules that were broken off from a seagrass meadow from something feeding or from a storm around Townsville could float almost the whole way up the Great Barrier Reef, almost to New Guinea. So there was massive connectivity potential there from just viable fragments of seagrass floating in the water. Given this is an ongoing process, there's millions of propagules out there all the time. There's always certainly going to be some that actually will settle. So Queensland's unique in that we have a southeasterly trade, which is why the wind currents will blow things to the north and the coast slopes back, so the things will float north. A key point here, though, is that the propagules move south to north. They're not moving north to south. And the southern part of the Great Barrier Reef is where everyone lives. So in some ways, what we do in the south could affect the viability and biodiversity of the north. We took that a little bit further, and bear with me, Alana and I did graph analysis or network analysis on those meadows. So we looked specifically at that Townsville region and how those meadows could interact together in terms of particles floating in the water. And we did a graph analysis where there's 33 meadows in that. The bigger the circle is, the more connectivity. The lines show where things are joined together. What Alana found is that the seagrass meadows around Townsville act as a small world network. They're incredibly well connected in terms of current movement. There are propagules moving between those meadows all the time. And we looked at measurements like betweenness centrality or out degree or in degree to see which meadows were well connected in that network. Now, this is the same as you might look at Facebook friends. You know, some people have got lots of friends, some people haven't. Some people are very close to their friends, some people have friends but they're not close to them. So a very similar analysis to that. And what it showed was that some meadows were very high in what we call betweenness centrality. There's lots of propagules moving in and out of those meadows. They're highly connected. If one of those meadows was damaged by something happening in the port or by a cyclone, it's very unlikely it wouldn't be receiving propagules back. It would be fairly safe from those changes. But other meadows at the edge of that network had a high level of out degree, were looking after themselves, were really robust, but not receiving propagules from the wider network. So we're able to look at different so actually then plot that network out on the actual coast, and you can see these meadows around Townsville are highly connected. There's also a really big connection between the deep water meadows and the inshore meadows, which meant if we had a cyclone, the deep water meadows tend to be more protected and they're likely to be okay. But they will then supply propagules to the inshore meadows, and quite strongly too. Now, this particular picture I'm showing here is only for the very small colonising species too. But it also meant that we could look at the edge of that network. There's an island bank here, so the network's quite limited. We could look at these particular meadows here, which have a high degree of high out degree, so they're providing propagules to the network, so they're very important, but they're not getting a lot of propagules back. Now, if you lost one of those meadows in a cyclone, it would be a meadow you might consider actually actively getting in there and restoring, because it may take a long time to come back. It's not well embedded in the network at all. And we're able to, if you read the paper, we're able to then actually physically take those meadows out of the network and see what effect they have. Because there's actually a port being developed at one of those meadow sites, and we're able to say, look, if you take this meadow out, it's actually very important to the network and could cause problems. But having said that, the network itself is very robust. You'd have to have a massive cyclone to actually stop that network moving. So we've got a world where the seagrasses are actually really, really quite connected through animal movement, through water current movement, through propagule movement. They're very robust to being... that's the end of me. I'll just finish off then, thank you. So in summary then, seagrass are a really important ecological community. They clean the water for you, they create biodiversity, and they do it all for free. Really important. They need light. These are plants. If you ruin the light, then you're wasting your time with anything else. There's no point trying to replant seagrass if the water's turbid. It won't work. The high energy locations are not good for seagrass. where you have big currents. You'll hear me say, if you live with me long enough, you don't get seagrass on a surf beach. They don't like high current stress. They live in places which are sheltered. That's also where you get ports and people living, because people like sheltered places too. Herbivory is a controlling element, and it's also very important, and it's also important to realise that a good seagrass meadow might be a very badly knocked around seagrass meadow because it's being eaten. The meadows are highly connected and quite robust, at least in our part of the world, and we'll extend that modelling out to the whole Great Barrier Reef. We just have to have a bigger computer because it's a big reef. And remember what I said, when you come to Australia and they say visit the Great Barrier Reef, just remember the Great Barrier Reef is the coral bit, which is really important. But inside that coral is a massive, diverse reef lagoon, which is really worth visiting. Thank you.
Thank you, Professor Coles. Since we run out of time, but I would like to allow only one short question. Is okay? Any question? Please.
Thank you, Rob. You've talked a lot about the importance of turtles and dugongs in regulating the seagrass beds. In this part of the world, Southeast Asia, there's been a massive decline in both turtles and dugongs, both accidentally caught in fishing nets and also illegally poached for food. Is there still a balance between the seagrass productivity and the abundance of turtles and dugongs in Australia? Or do you sense that the balance is delicate or even becoming an imbalance?
Yeah, really good question. It's really quite complicated to answer that because we have this anomaly in Australia where we live around Cairns, the numbers of dugong are going down quite a lot. We're supposed to be protecting them, there's no netting. If you go into Torres Strait in the north, where people are allowed to hunt dugong because it's traditional lands, the dugong seem to be doing really well. So we're not really sure what's going on. We think it might be a matter of just disturbance. So there's a bit of an emphasis on body count, but if you drive your boat up and down over a seagrass meadow so the animals don't feel comfortable feeding or calving, then you're just going to have a slow decline in that population. So I think it's a combination of netting, it's a combination of just disturbance, and a little bit of perhaps history there of what's happened in the past. We had a big shark netting arrangement in the country to protect people from being eaten by sharks, and that killed a lot of dugong. But we're getting better at not doing that, and the Queensland government's just banned netting in the Great Barrier Reef from 2027, which is a huge thing to do to the Australian fishing industry because it's very hard to catch fish without a net, on a fishing line. The turtles are slightly different. There's a lot of turtles out there, and I think eventually we're going to have a problem with too many turtles. Like, people want to protect turtles, but they're kind of not that short supply. And we know there's places around Australia where, there's an island called Cocos Keeling where the turtles are considered halal and they won't eat them. And we keep saying you need to eat these things. There's too many of them. Like, they eventually run out of food and they'll die anyway. So there's definitely an odd balance there that we're not really all that sure of. We've just done a complete survey of dugongs and turtles in the Great Barrier Reef, and that'll come out in the middle of the year and give us some idea how we're going. But the preliminary suggestions is it's not particularly well. The dugongs are going down in numbers in places they shouldn't be going down. They're oddly popular in places which there's quite a big population in Moreton Bay. For those who don't know, Moreton Bay is the bay right outside Brisbane, massive capital city. There's people everywhere, and that population seems to be doing okay. Around Cairns, there shouldn't be a loss of population, but there seems to be. In the Torres Straits, where everyone kills and eats them, they're doing really well. So I'm not really sure I can answer your question. I think we're going to have to watch this space and have to see how it goes. Like, I think it's a dynamic that we're not really sure we understand, compounded by the fact that once you get into the Gulf of Carpentaria, it's so expensive to go surveying anything, both in people's time and lives and money, that we really aren't able to give you the information on what's going on. We just have to do our best. We're using Aboriginal ranges a lot more. The Aboriginal, the lands up there are governed by ILUAs, or individual land use agreements, and traditional land use agreements, and they are coming on board now, and we're giving them advice, and they're doing quite a lot of the work for us, which will improve our knowledge in that part of the world.
So thank you, Professor Ho. Join me again to give a big crowd to him. May I ask Professor Somtak to introduce our next speaker? Yes.
I'd be honored to introduce our second speaker, invited speaker. Is I may say he's a young speaker, rising star, and he joined at the first IBD meeting. And I'm going to move you from marine environment to terrestrial environment. He interesting. The young rising star that I said is Dr. Chun Kobayashi. He's from the University of the Ryukyu, Okinawa. Interesting on pollination. As Professor Sanit entertained us this morning about the plant, the food, plant and food, and several plants came from the natural pollination. That Dr. Chun Kobayashi is going to give a talk on this. Dr. Chun he graduated in 2017 from University of the Ryukyu on pollination biology. He studied pollination, and since we know each other 10 years ago, probably, huh? Yeah, and we from time to time work together, and he published a lot of interesting on pollination. Pollination is very interesting for the ecosystem, biodiversity. Yeah, so let's invite, please, Dr. Chun Kobayashi for his interesting. Squirrel is an important animal that he found. Please welcome Dr. Kobayashi.
Okay, thank you for the introduction. Good afternoon, everyone. So at first, thank you for inviting me as a speaker in this conference. I appreciate all organizers. So this is a great opportunity for me. So the topic of my talk is plant-animal interactions, especially focusing on the pollination. So the topic of my talk is very specific compared with other keynote speakers, but I think it's interesting. So let's start my talk. So the pollination ecology is one of the research topic of plant speciation, but it's also the topic of coevolution of animal and plants. So I'm studying this phenomenon in the coevolution point of view and studying based on the field surveys. So anyway, nearly 300,000 plant species are known about, and about 90% of angiosperms are pollinated by animals. So therefore, angiosperms diversified by relying on animals for pollination. So these photos show the pollinators. So the most abundant pollinators are insects like that, and especially bees. However, there are other animals considered as pollinators. For example, land snails, hermit crabs, geckos, birds, and some other mammals. So in my talk, I focus on mammalian pollinators and mammal-pollinated plants. So at first, I'll introduce you about the outline of mammal-pollinated plants. In the world, mammal-pollinated plants are estimated as more than 600 species, and that is only 0.3% of angiosperms. So this table shows the proportion of pollination mode in tropical regions. The target species are only 3 species. So for example, in Kakachi in India, 86 species, 3 species are surveyed, and only 3% are pollinated by mammals. And in Rambil Hills in Borneo, 127 species are surveyed, and only 4% are pollinated by mammalian species. So, unfortunately, my target group is very minor. Among mammal-pollinated plants, bats are well-known pollinators. So I'll talk about bats. So bat species, all bat species are known in 1,000 bat species, and including 200 nectar-feeding bat species. So nectar-feeding bats means nectar-feeding specialist and not specialist. And not specialist means fruit bats. Fruit bats eat some plants, some plant materials. For example, not only the nectar, but also the leaves and fruits and so on. So when nectar-feeding bats feed on nectar, pollen grains attach to it. So this is a good example, the pollen attached to the face of bats. So nectar-feeding bats are considered as pollinators. So I introduced you about the roles of fruit bats. So the nectar-feeding bats, especially the fruit bats, have some roles for ecosystems and for humans. As for the ecosystems, more than 500 plant species are pollinated by nectar-feeding bats. In addition, many seeds are dispersed by fruit bats because some of them feed on fruits. So such roles are important for humans too. Some agricultural products, such as durian, palkia, and kapok, are mainly pollinated by fruit bats. So these fruits are our food and life materials, and also they provide us with income. So the fruit bats are very important for us. Among all mammal species, bat species are only 1/6, and among them, about 200 species contribute to pollination, as I mentioned before. Then, how about remaining 5/6 of non-flying mammals? Do they pollinate some plants? Actually, I researched on non-flying mammal pollinated plants, or not so many. So next, I roughly compare, wait a moment. Okay, so these are non-flying, example of non-flying mammal pollinators. So in 1997, one review paper published. It's more than 20 years ago. So after that, no review papers are released. So there are not so many researches. So these are examples of non-flying mammalian pollinators. So, okay, so this is possum, rats, civet, masked palm civet, elephant shrew, lemurs, and tree shrews. You know some of them, I think. So next, I roughly compare bat-pollinated plants and non-flying mammal-pollinated plants. So this figure shows the phylogenetic tree of angiosperms. So colored line, like these, show the plants, including bat-pollinated plants. So the bat-pollinated plants are polyphyletic and recorded from various families. In addition, there are regional characteristics. In some families, bat-pollinated plants appeared only in the New World, but in other species, the bat-pollinated plants appeared only in the New World. In addition, in several families, bat-pollinated plants appeared both in Old and New World. And then, non-flying mammal-pollinated plants are surrounded by the black line, like this. like this. So non-flying mammals, non-flying mammal pollinated plants appeared in various families like that. And it's also polyphyletic. And the next. So then I summarize the pollinator of bats and non-flying mammals. So taxonomic group and flying abilities are different, of course. And relating to the flying ability, moving distance in a short time also different between bats and non-flying mammals. So bats moving distance in a short time is long, but non-flying mammals are short. In addition, their food habits are also different. Bats are herbivore, as I mentioned before, but most non-flying mammals are omnivore. So the non-flying mammals also eat animals too. Those differences of pollinators may cause some differences of plant characteristics. Furthermore, relationships between plants and pollinators are different by regions. So the Wilma 2011 shows the summary of pollination system of main genus of plants, including non-flying mammals. So 4 genus are shown in Australia, South Africa, Costa Rica, and Brazil. For example, in Australian Banksia, the non-flying mammalian pollinator, the marsupials, for example, possum and corals and so on. And the plant characteristics are shown in the bottom of the table. And the floral color is pale yellow and brown, and the position is hidden and low. And the nectar secrete nutrition-rich nectar, the floral scent is weak to strong, and the flower position, flowering time is at night. And in South African Protea, the pollinators are different from others, and the main pollinators are mouse and elephant shrew. But the floral color and other characteristics are similar with Australian species. But in this table, there are no Asian species. So how about the Asian species of non-flying mammals? And actually, at that time, there are only a few researches in Asia. That's because I didn't start the research. So after that, several papers are published from Asia. So I reviewed published papers and tried to summarize the trends of non-flying mammalian pollinator and non-flying mammal pollinated plants. So I searched papers by using the Web of Science and Google Scholars. And in this study, pollinator was defined as a species with pollen attached to it. Eventually, I found 22 relevant published papers recording non-flying mammalian pollinators and non-flying mammal pollinated plants. It means that research is still not enough. Anyway, this is the distribution map of non-flying mammal pollinated plants in Asian regions. So non-flying mammal pollinated plants were recorded from tropics to temperate regions. And many are recorded from Southeast Asia. And then the next is pollinators. So this table shows the number of pollinators in each family and order. In total, 33 non-flying mammalian pollinators have been recorded. Among them, squirrel families are the most recorded group. So there, and this is very interesting compared with other regions. Okay, so the next is the focusing on this subfamily. So about squirrels, number of species in subfamily Carolina is the highest. Those squirrels are diurnal. In other regions, squirrel pollinators are rare, and many other pollinators are nocturnal. So this is also the characteristics in Asian regions. As for this subfamily, this group has some evolutionary interest. So first, this figure shows the phylogenetic tree of squirrels. So this subfamily is categorized to clade 3, this one. So this taxonomic group has been diversified in tropical Asia and includes 64 species from 12 genus. And the second, these squirrels feed on nectar. However, many other squirrel species do not feed on nectar so much, so it's a very interesting thing. In addition, these squirrels use various environments. Therefore, squirrels in this group have the potential to contribute to pollination in a variety of environments, from forest to urban environments. Okay, so the next is the plant characteristics. These are examples of non-flying mammal pollinated plants in Asia. As you can see, floral colors, shapes, and sizes are varied. So this is a list of plants, those pollinated by non-flying mammals. So Fabaceae is the most frequently recorded family, but non-flying mammal pollinated plants are recorded from various families like that. In addition, much of them are tree and climbing vine species. In other regions, much of non-flying mammal pollinated plants are shrub or low trees. So it's the interesting thing in this region. Furthermore, most species are pollinated by squirrel, and squirrels frequently use trees, so it may relate to growing type of plants. And among the Fabaceae, several non-flying mammal pollinators are recorded from genus Mucuna. So from here, I'll try to fill the gap of regions by using genus Mucuna and compare the pollination system among regions. So these are Mucuna flowers. And this genus includes more than 100 species, and several of them are used for medicine. Origin of this genus is in tropical Asia, but this genus is recorded throughout the world. Flowers are large and colors are varied, like these photos. And before talking about pollination system of this genus, I talk about the shape of this flower. So flower shape of this genus is called papilionaceous flower. In other words, keel flowers or butterfly-like flowers. So one of the interesting things in this genus are the pollination mechanism relating to the shape of this flower. Okay. So this is a matured flower. So flowers are constructed by 5 petals, so banner petal, a pair of wing petal, and a pair of keel petals. Interestingly, you can't see the stamens and the pistil from this photo. The stamens and pistil are important organs for pollination, but these organs are hidden by keel petals here. So if nothing happens, a flower will drop off and it cannot bear seeds. However, when a flower opens like this, stamens and pistil are exposed from the keel petals. In this phase, it can pollinate. So this flower opening step is called explosive opening. I'll show you the video, a slow motion video, but it's not working. Okay. Sorry, not working. Okay, so just pass. Okay, so pass. That's right. Actually, the pollen released explosively, so we called this step explosive opening. And then I checked the flower opening power. It's very interesting thing, but if you can not see the video, it's difficult to understand. But I explain about that. So I actually recorded the flower opening force of mucuna, genus mucuna, and compared with the plants pollinated by bees. So these are flower opening force of these species. And the flower opening force is quite larger in mucuna species compared with bee-pollinated plants. It means that the pollinators are limited to large animals. So I'll show you some video of the pollinator, but I think it also doesn't move. Okay. Pass. So this is another example of a squirrel. So this one is a variable squirrel in Thailand. So I took this video in Thailand. So the squirrel opened the flower very skillfully. So the squirrel holds the wing petal, this petal, and purple one, and then push up the banner by its nose. So it's very skillfully and open the flower. And next example is this, and this is musk palm civet. Oh, thank you. Civet open the flower like that. So musk palm civet also holds the wing petals and then push up the banner by its nose. So the opening behavior of musk palm civet and squirrels are the same. Oh. Doesn't work. Sorry, next example is the actually monkey. The monkey is also the pollinator of mucuna species, and the opening behaviors are different from others. So yes, video. Yes. So it's open by using only both hands. So macaque hold wing petals by one hand and open the flower by another hand. So the opening behaviors are quite different. Okay. Oh. Okay, okay. Actually, so these are plants with confirmed pollinators in Asian regions. So although one species is pollinated by only birds, this one, but in other species are pollinated by non-flying mammals such as squirrels, monkey, civet, rats, and tree shrews. So furthermore, some species have both diurnal and nocturnal species. Okay, then the next is a nectar and floral scent. So left side figures shows the nectar secretion pattern of two mucuna species. In mucuna, flower blooms throughout the day, and nectar is stored in all the day, even though the bud-pollinated plant. So, and right figure shows the result of a principal component analysis of volatile components of 7 mucuna species. Each character shows the species. Although mucuna flowers emit fermented smell, this figure indicates that volatile components differ among species, and those do not have trends relating to the pollinators. So from those results, I summarize the pollination system of mucuna and compare with other regions with other genus. First, in the pollinator. So the taxonomic group of pollinators of mucuna are different from others. Some are pollinated by squirrels, civet, and tree shrews. In addition, non-flying mammals are both diurnal and nocturnal, while pollinators in other regions are mainly nocturnal. So the next is the plant characteristics. So the characteristics of floral colors and scents are not unique in mucuna species. However, nectar secretion pattern and flowering time are different from other regions and other genus. In other regions, flowers bloom at night and secrete nectar at that time. But mucuna flowers open several days and nectar is stored throughout the day. So it may be related to the activity pattern of pollinators. So this is the conclusion of my talk. So at first, various taxonomic groups of non-flying mammalian pollinators have been recorded in Asia. And among non-flying mammalian pollinators, squirrel are the most recorded non-flying mammalian pollinators. Therefore, squirrel are the representative non-flying mammalian pollinators in these regions. In addition, taxonomic group and lifestyle of non-flying mammalian pollinators of mucuna are different from pollinators of other genus in other regions. These are unique relationships in these regions. And finally, studies are not enough in these regions, and much more non-flying mammal pollinators and non-flying mammal pollinated plants are available. Researches on plant-animal interactions in Asia will be developed, and the other characteristics of evolutionary trends in Asia might be found in the future. So I'll continue the field survey about plant-animal interactions, and I hope to be able to share my future results with you. So thank you for your attention, and Koppen Cup.
We still have 2 minutes, so 2-3 questions are welcome. So please.
Kopkunkat, one quick question. Are any trees exclusively depend on the squirrel for pollination?
Pardon?
Any trees that you know that must have the squirrel to do the pollination for them? Exclusive. Thank you.
I didn't conduct exclusion experiments, but several of them visit only squirrels. So the squirrels, some plants are very important pollinator.
Kopkunkat.
Please.
This is the first time that I ever listened to the good point of this small animal squirrel. Because in Thailand, in almost our house garden, we can find squirrel. I just would like to ask you, have you ever had any information about any toxic substances or any disease transmitted by this small animal?
I never heard about the toxin in that animal.
You have never done or interested on this
Never.
subject?
Never learned.
Since you are interested to study more on this small animal, would you please consider to study on this? Because
thank you.
we have quite a lot in almost every house. Thank you.
Thank you very much for the comment.
Okay, I think we no more questions, and it comes to the 30 minutes time. So, okay, thank you. I would like to make a big applause to you to all. Dr. Kobayashi, thank you very much. Yeah, with the chairman.
May I invite our last invited speaker, Dr. Vichan Simachaya. He is the President Board of Thailand Environmental Institute and Secretary General of Thailand Business Council for Sustainable Development. He will talk about the role of private sector in contributing to biodiversity conservation in Thailand. Ajahn Vichan, please.
Thank you very much. Good afternoon, everyone. I'm not talk about species. I'm going to talk about the role of business contributing on biodiversity conservation in Thailand. I myself, President of TI, Thailand Environment Institute. At the same time, I also serve as Secretary General of Thailand Business Council for Sustainable Development. My talk going to cover the policy, especially related to the biodiversity. You can, maybe you know already, Professor Sadit already mentioned about SDG, Sustainable Development. We can take a look what is SDG, especially SDG related to biodiversity at the SDG 14 on the right below water, especially for the coastal and marine resources, and SDG 15 life on land. We talk about tourism ecosystem, wetland already include. You can take a look how international organization evaluate our SDG in Thailand. We pass to SDG. The first one is no poverty. The second one is the quality of education. But some people may have a question by yourself. Why the quality of education already passed SDG in the green zone already? But when we take a look at the biodiversity, especially the 14, the coastal and marine resources, and 15 is still very dangerous zone at the red zone, especially for the 15, and dangerous zone, red zone, and go down. If we not protect right now, how we can reserve our natural resources to our next generation? This is the Thailand. Thailand, we have a lot of plans. Actually, we have long-term strategy, 20 years. We have national economic social development plan, even our new national agenda on the BCG, Bio-Circular Green Economy, also addressed on the how important of the bioeconomy that already included, but how we can work, especially implementation of the biodiversity conservation on the ground. I think it's a really important issue. Biodiversity is the cost-cutting issue, especially interlinked with the various significant concerns, especially associated with the SDG 17 goal. I already mentioned this is how important of the SDG 14 or SDG 15 related to biodiversity. How we consume our biosphere, you can take a look. What the scientists say that we already spend our Earth more than the Earth provides to us, 1.7 Earth already. You can take a look, capacity deficit for the year 2018 in the red zone, especially for the developed country like the US, China, Russia. But in our country, in Thailand, we already consume 85% of biocapacity. This is the important issue. You can take a look. This comes from the World Economic Forum for Global Risk Perception for the year 2022 to year 2022. You can take a look. The green color is show the problem of the world in the next 10 years. You can take a look. The first 4 related to the environmental issue. The first one is the mitigation to the climate change, especially the problem that you may know already. We talk about net zero. We talk about carbon neutrality. How we get there is very important. We set the target of the goal of the country, of the global, how we get there. And the second one is the adaptation. Also very important. The climate change impacts are really going to occur. We are facing right now in various regions of the world, but how we can adopt ourselves resilient also very important. The third one is the national disaster already occur in many areas in the world, like heat wave, very hot in the cold region, very dry in the wet region or something like that. The flooding all over the world already occur. You can take a look at the fourth one is the biodiversity loss and ecosystem collapse. That's very important. If we can compare to the web, you can take a look. The COVID-19 almost gone, but what they said, COVID-19 is really small symptom for our world. But the bigger one is like the economic recession followed by the climate change problem. I myself just come back from the COP28 in Dubai, talk about the mitigation of the climate change. At the same time, we also talk about the adaptation. This is the first COP climate change. They talk about the most country around the world talk about the linkage between the climate change and the impact of biodiversity, especially for the biodiversity collapse, biodiversity loss I mentioned earlier. Business activity. I myself, I already mentioned Thailand. We have Thailand Business Council for Sustainable Development Organization that join hand together with the private sector and the government sector as well, try to work on the environmental and social issue. Biodiversity is the one issue that brought to our committee. When we talk about how important of business on biodiversity, we have to look raw material, especially some business they related to the food, to the ecosystem, to the natural resources, transportation, over exploitation, processing, and wholesale, even retail services are also very important issue that link to the pollution related to the impact of the biodiversity as well. We talk about alien species as well. Business dependency on nature and biodiversity. Just why I myself serve as Secretary General, we brought the issue to the business forum. At the first stage, very difficult for myself because a lot of company or even member, they just heard the word of biodiversity for the first time. I have to explain to them what is biodiversity. That's the education awareness also very important for not just solely for business, but for the people in general as well. This is how it links business and biodiversity. We have our national agenda on biocircular green economy that based on the principle of sufficient economy, a philosophy of late King Lamanai, and also join with the support with the Sustainable Development Goal I mentioned earlier, but we have to look back at ourselves, what we have. We rely on biodiversity. We talk about food security, health security. We have herb, energy security, especially energy in transition for the bio-energy as well. Employment security. Last not least, very important one, how we can sustain our natural resources and environment. His BCG action plan already mentioned the utilization of bioeconomy. If you would like to know more, you can search and look for more. Business also work on what we call ESG. They have to submit the annual report, environmental, social, and governance. In the environment right now, environmental issue right now, they concentrate on the greenhouse gas, especially carbon dioxide reduction. Biodiversity is a new issue that we just start to talk about it. That's the situation of business and biodiversity in the country. I myself start to talk with the international organization like the government of France, also interested to support Thailand on the how business work on the environmental issue. This is they have some fund, they have some green fund, green bond that already mentioned about that. But how we can cooperate with the Thai policy, I think is very important. For example, we talk about the tourism or something like that, that we sell our natural resources, we sell our biodiversity, how we utilize them, how we conserve them for the sustainable policy. I think it's a very important issue as well. That's the example that the local people, they protect the forest, why they can grow like a coffee tea under the tea, that they also conserve biodiversity at the same time as well. That's the example of Got Tao in the southern part of Thailand. They protect ecosystem, marine ecosystem, especially for the reduction of the plastic pollution, while we can promote the tourism as well. This is we also start with the to work on the supply chain, especially for the palm oil alliance. Just start in our country. Plan the palm oil, how protect the biodiversity in the farm, something like that. That's also the new agenda that we are working on. And strengthening business biodiversity is really important. We have to focus on the area, for example, food sector. We start to talk about the company that work on the food sector, tourism sector, that Bedo also promote biotourism as well. Start from the company. Some company already get started with the biodiversity conservation concept, quantify value in the monetary term. I think it's really important for the business people driving BCG as a national agenda. Here just show you the value of biodiversity, our challenge, our way forward. We have some tools, for example, like a PS, payment for ecological services. Even Bedo also work on the BB check, the business and biodiversity check, and national capital accounting, something, and also related to the biodiversity. Is the key takeaway. Natural loss is the accelerating. I showed you, talked to you earlier, the impact of biodiversity, especially from the climate change, the list of business and finance arising need an integrated approach, because the biodiversity, we have to look for the ecosystem. Pollution also one of the case impact to biodiversity, dependency, impact the least opportunity. I think that kind of thing we have to consider. This is the climate change and biodiversity is related. How important of biodiversity, how we can protect, especially impact of the climate change, high temperature, low temperature, for example. Finally, I would like to introduce you. I mentioned earlier, TBC, Thailand Business Council for Sustainable Development. We are 45 organizations in Thailand. And you know, if you want to know more, you can take a look on the second floor. We also have our exhibition, what we work on the biodiversity issue, business for biodiversity. That's new issue that we bought BB Check from Bedo that promote also to the member of TBCSD. That's the example that our member like Bangchak also develop the step of the development of biodiversity in the business. Start from the commitment, especially policy is very important, and risk assessment, monitoring, and mitigation action. What action do to protect the biodiversity loss, and tracking, monitoring is very important issue as well. And the communication with the company member, with the employer, with the community nearby, the factory, I think also very important issue. This is example, they work on the 5 conservation project at Kung Bang Kachao, Samut Prakan province, that green area in nearby Thailand. Also with the Prang Yai, Ban Nam Phung, Bang Kachao, also conservation of the bee, and come to the local product. They do also support like bio product. It's mostly right now we work on net zero, but how to incorporate to biodiversity, I think is a really important issue that our message to bring to the business forum. Act today, I think is very important for a better future and for conservation of our biodiversity. Thank you very much.
So floor is open for questions and discussion, please. And any questions?
Good afternoon, sir. Thank you for your lecture. I just want to ask, because you're talking about the business sector, so is the companies, do they have research and development, or are there any collaboration with the academe with that for studying biodiversity?
Yes, I would say yes. Actually, the company, they're not researcher by the company themselves, but they hire the consultant, especially from the researcher from university. I talk about the monitoring system also, vertebrate-based monitoring impact to the ecosystem or something like that. They have to hire the specialist on that issue, then they make the report for them. I mentioned earlier for the listed company that they have to support, we call one report. One report cover to the financial sector, cover on the ESG, environmental, social governance, but environmental issue have to cover biodiversity issue as well. But in detail, we have to work more with the stock exchange, that office, how they consider on the list of biodiversity. The company have to consider on that. For example, the construction company and talk to me what they can do with the biodiversity. I also give them some very simple one. A very simple one is when you the environmental impact assessment or EAA report, they have to consider on the green area. They have to plant some plant, but when they're going to bring some plant in, they have to consider on the local plants or something like that, and how to conserve nature that the area. If the construction is close to the canal, they have to taking care of the ecosystem of the canal as well. This is the example. I can talk to them, easy example for that. But more detail, they have to work closely with the researchers from various universities.
Okay, sir. Thank you very much.
My pleasure. Thank you.
Any more questions? Oh, many. So,
Thank you, Dr. Vijan, an excellent presentation. I have a very basic question. What do the typical members of the Thai public understand by the word biodiversity? Because in my country, Scotland, we asked farmers what did they understand. And the answer was something about birds.
Something about art.
About birds.
Oh, about birds. Okay. Similar to our country culture. I used to work from the Minister of Natural Resources, the previous minister. What the heck of the biodiversity? What he said? I just heard from the first time of my life, almost 68 or something like that. That's the Ministry of Natural Resources said that. I have to explain to them what is the meaning of biodiversity. Bio is mean like life. Everything is allow you with the life and ecosystem or something like that. But when we talk about ecosystem, we have to explain more what is the ecosystem. That's the, I think I have to Professor Napawan also. She teaching for a long period of time how teaching the student to understand the biodiversity. This is the important issue. The awareness, education, I think is very important. When we give the information to the people, to the student, even you talk about the farmer, right? We have to keep the simple one, especially their experience. If they work on the paddy field, what is the biodiversity in the paddy field or something like that? That would be good example. It takes time.
So you want to, yes, one last question, yes.
Okay, yes.
So thank you for that wonderful presentation. I have a basic question. Are you the operator for the Global Environmental Fund? And maybe if you catch that, what will be the policies in Thailand if Thailand will get a Global Environmental Fund for an extra round? What are the main lines for using this kind of global funding?
Yeah, you question about the funding, right?
Yes, Jeff. Actually.
Yeah, we have a very sort of fund. I myself also work with Professor Napawan on keep the funding to researcher, national funding researcher. But the problem is that we can find a good researcher to keep the fund. That's also the problem. And we have the fund from Thailand. We also have, we call environmental fund. Also have some fund that the researcher can act to support the research on the biodiversity as well. And the international funding, we just start to talk to the AFD, maybe from funds, government, to support the business on how important of business for biodiversity sometimes. That's the really early stage for Thailand. And we give the Sobedo support, give the support from JEP like Global Environment Facility on the biotourism development for the country.
Okay, thank you.
Okay, thank you very much.
Thank you very much. Give me a Thank you.
Okay, now we will have a 15-minute break. Please be on time for 15 minutes. Let's say 4:30, we will begin the second half of the first session, oral presentations. Thank you. our distinguished guests, ladies and gentlemen, let's come back to our second half of the first session. We will start with the first group of oral presentation, and please welcome back our chair and our rapporteur, Ajahn Savitri and Ajahn Somsak, please. Thank you.
So let's start we will have 5 oral presentations. So may I invite the first presenter, the first group of presentation, the topic of their talk, because I say their talk because they will have 3 speakers. The title is Taxonomic Revision of Genus Balanophora with a New Record from Thailand. Please.
Thanks for introduction. Greetings, ladies and gentlemen. Today, we are going to present our research, Taxonomic Revisions of Genus Balanophora with a New Record from Thailand. Our group members include
ณัฐพฤกษ์ ศักดิ์ไร้พานิช ธนเศรษฐ์ กิติวัฒนไทย
and Sithiporn Singnalinthorn. This work is under supervision by advisor Dr. Tiro Sang Ratanaprasad, and here are contents. 3, 2, 1, next slide. First, I would like to inform everyone about the taxonomic information about Balanophora genus. Well, Balanophora belongs to phylum Kingdom Plantis in order Centellales, and they have generic type Balanophora fungosa JR4 and G4. These plants were grown in tropical to temperate zone. So from the distribution map in the bottom right of slide, you can see that it's commonly found in Southeast Asia. In Thailand, you can find it in tropical rainforest and mountain forest, which are suitable hosts. And a whole genus of Balanophora is holoparasite, means that it can't photosynthesize by itself. That's why it must gain nutrients and water from the host plant. And as of 2023, there are 25 species accepted by Plants of the World Online. Next slide. Well, from the appearance of Balanophora, you see that it looks like mushroom. So someone might be misunderstand it's a fungi, but actually it's a plant. And Balanophora is come from Latin, means acorn bearing. So from the picture, you can notice this. The shape of Balanophora is like the upside down acorn shape.
So now let me show you about benefits and impact. Take a look at this following slide from research of Mr. Chai Viratna et al. In 2020. The insects are pollinators, which the most species of pollinators belong to order Hymenoptera, such as Apis cerana, and Anopolypus gracilipes are this. And in 2023, Suesuku and Hashiwaki, they conducted a study in the understory of dense forest on Amami Oshima. They used infrared cigarette cameras to observe Amami rabbits' behavior. And they found that Amami rabbits ate Balanophora yuanensis fruit. And so the Amami rabbit is this dispersers of Balanophora yuanensis fruit. Balanophora fungosa is also an indicator to indicate the abundance of environment. As the work of Dr. Hanjai et al. In 2011 studied ecology and biology of Balanophora fungosa at Doi Sonon, Chiang Mai province. For the utilization, The tubers of some species contain wax-like substance which Javanese people use for making torches. Next, we are going to talk about our methodology. For literature review, we had read up in order to find out the information about Baron Flora from many sources, such as Flora of Thailand, Volume 2, 1972. It provides us taxonomic data of 5 taxa of Baron Flora in Thailand. And however, we also used Hansen of the genus published by monograph of Hansen, published by Hansen for identification. We also share our taxonomic information from websites which collect plant taxonomic data around the world. Those are Tropicals. Those are Tropicals, IPNI, and Plants of the World Online. We study herbarium specimen deficit in many herbaria like BK, BKF, and CMUB at Chiang Mai University. We observe Baron Flora in its natural habitat as well. And now we are at this stage, species conclusion and description. We are continuing to summarize how many species of Baron Flora in Thailand, and we combine the information from our herbarium and fresh specimen from field trip. Then plant description and key to species were constructed.
Now it's time to talk about the morphology of Balanophora. Well, some of this morphology we used to create the dichotomous key in the end of our project. Okay, first, Balanophora can be divided into 2 parts. First is tuber. Tubers of Balanophora is like potato tubers surround the host root. Normally, we found it surround the young host root and suck the nutrients and water from the host plant. Next is inflorescence. Well, Balanophora inflorescence can be divided into 2 types, dioecious and monoecious. And when we zoom into the inflorescence, we will find the forests. The top of slide is male forest. For the male forest, we have a synandrium in the center. It's actually the fused anther. And we have the tepals around it to protect the synandrium. And the bottom of the slide is female forest. Well, the female forest is massively reduced into just 1 part, which is the pistil. And we have another part of Balanophora called the spedicle. Actually, it's a reduced bracts to protect the pistil. Well, the positions of pistil which stick with the spedicle can be used to create the dichotomous key too. And the last one is the pedicels. The pedicels can observe in only the male inflorescence, and the length of pedicels also can use to create the dichotomous key.
So after we have observed the morphological characteristics, we can finally create a dichotomous key, as shown here. So this is the one that we have created using various characteristics. For certain characteristics, we include our being monoecious, dioecious, male floret symmetry, and male floret's morosity too. The leaves arrangement, leaves color, and the general shape and size can also be a determiner for the species itself. So here are all the listed taxa currently in Thailand as of 2023. The first one is Balanophora abbreviata. This species is a monoecious taxa that, as you can see in this picture. On the top is the female inflorescence, and on the bottom are the male inflorescence. Its unique characteristics of the species is that the male inflorescence, male florets specifically, are zygomorphic. And the pedicels, the stalk basically of the male florets, are extremely short. And the location of the species itself are mostly found throughout the northern regions of Thailand and also sometimes in the south. The next species, similarly to Abbreviata, is Baronophora subcupularis. This species differs from the last one by being actinomorphic for the male florets and the leaves being more reddish and distichous. The pistils in the female inflorescence also occur at the base of the spaticles, and there's also a more whitish color instead of the creamy yellowish colors of Baronophora abbreviata. The distribution of this is mostly on the northern Thailand side, with some on the northeastern side. Next is Baronophora hollandii. This species differs from other taxa in Thailand by being in the subgenus Balania. The species specifically is the male florets are actinomorphic with 3 merous flowers, so there are 3 tepals on the male inflorescence. The female flowers are on the pedicels are on the center, and the leaves are subopposite or spirally arranged. It can be found mostly in northeastern Thailand and sometimes in the north. Next is Baronophora fungosa subspecies indica. This species is the most commonly found in Thailand and one of the most widely seen. It is a dioecious species with actinomorphic male floret symmetry. Its male florets are 6 merous usually, with pedicels being unusually long. It can be found almost throughout Thailand except for the southern part. Next is Baronophora laxiflora. This species is a dioecious species that can discern from other species by being the female inflorescence being really oblong and elliptical. Its pedicels are really extremely short, with male florets being 6 merous. And as you can see in these pictures, herbarium specimens, the female florets are really long. The leaves and the colors of the stalks are dark to crimson red and also almost turning purple. Lastly is another species that is often mistaken for Baronophora laxiflora, which is Baronophora laticephala. The species is also dioecious, and the female inflorescence itself is also quite elliptical. What differs from the last species is that the male florets, as you can see in this picture, are 4 merous and more zygomorphic. The leaves are also yellowish to whitish yellow, and the pedicels are also a little bit longer. It can be found almost throughout Thailand and usually in the central Thailand region to the northern region. While we were researching these projects in various herbariums, we also found a new discovery, namely a new record for Thailand, which is Baronophora papuana. And this species is unique from other species by various characteristics. For example, the tuber surface, which is the surface of the underground stem, is mostly polygonal verrucous, and the female inflorescence are also more ovoid. Its main characteristic, though, is its pistil occurrence. Other similar species like Baronophora fungosa species indica or Baronophora hollandii, the pistil occurs usually on the cylindrical part or the central axis of the spaticles. Instead, this species, Baronophora papuana, is mostly situated on the base of the spaticles. This species was originally found in southern Thailand, and as here. Here are the illustrations of the specimen in question and the specimen in the herbarium. And we thank you everyone for letting us speak here, and thank you for listening.
So welcome for one short question, please.
Your performance and your study research is excellent for high school. I think your work is like a PhD, study, and I hope in the future you will be one of really important or famous taxonomists please go continue to study at Chulalongkorn University. For the student of Dr. Somsak, well, I don't want to jump it, you know. But one thing that is wrong for the picture, you know, that you said about the honeybees. But the picture is not an epicellina. You should go to, you know, like Professor Chan Pen or somebody, taxonomies of honeybees in Chulalongkorn University. I mean, a bee biologist. The picture that you show is Vespa, you know, it's wasp, you know, not epicellina. You know, just one that I can see, but it's not important for your new record of this species. Okay, so in the future, try to study on more detail than this. Thank you very much.
Thank you very much.
So thank you. Join me to give a big round of applause to them. Thank you very much. Yes.
Okay, we go on to the second papers by Dr. Tricia Joy C. Torres to the benthic macroalgae of Eastern Siakau Island, checklist diversity, abundant distribution, and community structure. Yes, please welcome Dr. Tricia.
Hello, good afternoon, everyone. Just a correction, I'm still a masteral graduate student, so I'm still not yet a doctor, but maybe someday. So, again, I'm Tricia Joy Torres, and I am a graduate of Mindanao State University Iligan Institute of Technology. And the title of my paper is The Benthic Macroalgae of Eastern Siargao Island, Species Composition, Diversity, Distribution, and Community Structure. So, macrobenthic algae are photosynthetic marine plants, mostly found in marine, brackish, and freshwater environments. So, they grow well in rocky areas, especially they are attached in dead corals and rocks. And they are distributed from the low intertidal to high tidal zones. And as what Dr. Coles, the first speaker, said, seagrasses have true root systems, right? But in seaweeds, they have no true root systems. Instead, they have attachment organs called holdfasts, which is similar to the roots of terrestrial plants. And they also have a stipe instead of a stem, which forms into a lamina, which looks like leaves. Now, macroalgae are divided into three main groups. So they are the chlorophyta, ochrophyta, and the rhodophyta. So they are grouped based on their chemical composition. So the chlorophyta is when the dominant pigment is the chlorophyll A and the chlorophyll B, and the ochrophyta is when the dominant pigment is the xanthophyll and beta-carotene, while the rhodophyta is when the dominant pigment is the phycoerythrin and the phycocyanin. So the presence and distribution of macroalgae are influenced by a variety of environmental factors, such as the activities in the local communities, as well as the tourists and the physical and chemical properties of water. So these marine plants play a vital role economically and ecologically. They interact with other components of the ecosystem and are contributors to the primary production, nutrient binding, and habitat formation. These plants are of high value in economic industries since they are also the sources of the three main hydrocolloids, and these are the agar, carrageenan, and alginates, which are the three leading seaweed-derived hydrocolloids. So alginates are used in paper textiles, Agar is mostly derived from the red seaweeds, and they are used globally for biotechnology and pharmaceuticals. Carrageenan can be isolated from the red seaweeds, such as Kappa ficus, and they are used also in many biotechnologies and pharmaceuticals. So macroalgae are also a source of the phenolic compounds such as the saponins, flavonoids, alkaloids, and tannins, which are the most common secondary metabolites. And they are of great value in medical industry since they are used as antioxidants, antibacterial, and anti-inflammations. Seaweeds also hold not only in biotechnology and pharmaceuticals, but they are also a source of food. So especially in the Philippines, we have a lato and a guso, which is a very famous delicacy in the Philippines. It is used as a side dish. So similar to other living taxonomic groups, the demand for macroalgae biodiversity studies is increasing and work is ongoing to new taxonomic information. So currently, there is a roughly 11,000 species around the world, and the leading group of macroalgae is the red seaweeds, followed by the brown seaweeds, and lastly, the green seaweeds. So there was a trend wherein during the 1990s, the shift in macroalgae study turned from biodiversity to biotechnology. So today, though there are still many studies in the biodiversity of seaweeds in the Philippines, there is still a lot of needed baseline information for macroalgae. So the objectives of my study is to determine the benthic macroalgae in 8 sampling stations around Siargao Island, Surigao del Norte, in terms of its species composition, distribution, diversity, and community structure. Second is the result of this study would broaden the geographical distribution of macroalgae in the Philippines and it would serve as baseline information for future biodiversity research of this important marine resource. And the most important thing is that no study has been conducted on the macroalgae in Siargao Island. So this is the first study for the macroalgae in Siargao Island. Therefore, the data and other information generated from this study is very timely in order to promote the essential role of macroalgae or seaweed in the food security and sustainability and their effects on the environment and economy in this beautiful tropical island of Siargao. So here is the map of the sampling site, or the Siargao Island. So Siargao Island is situated geographically and politically in Surigao del Norte, which is the northeastern part of the Philippines. So Siargao Island is very well known as the surfing capital of the Philippines and one of the great frontiers for surfing. So the scope of this study is to determine the species composition, distribution, diversity, and community structure of benthic macroalgae in 8 sampling stations. And it also focuses on the protected landscape, seascape area abundant in marine sources. So the environmental parameters were limited to water temperature, salinity, pH, dissolved oxygen, and water flow, and the measurement was done once, and that was during start of the sampling. So the identification of macroalgae was based on external morphology using existing taxonomic keys and published journals. So the materials and method is the establishment of transect line, wherein I used 3 transect lines perpendicular to the shore with 5-meter interval of quadrats in each transect line. So there is a total of 10 quadrats in each transect. So times 3, so there is a total of 30 quadrats for each sampling sites. So the collection of data, it includes the physical-chemical parameters, the measurement of frequency, density, and cover of seaweeds, which is based on the methods of Saito and Otobe, the collection, identification, and verification of benthic macroalgae, and also, I forgot, the measurement of diversity and community structure was based on a lot of authors, Lee et al., Margalev, and Odom. The diversity and community structure, these are the IVI, or the Importance Value Index, and the diversity indices. And for statistical analysis, PAST was used for the statistics. So the results and discussion. So the present study enumerated a total of 62 macroalgal species comprising 29 species under chlorophyta or the green algae, 18 species under the phaeophyta or the ochrophyta, which is the new phylum for brown algae, and the 15 species under rhodophyta. So these are the pictures of my green seaweeds. And now the brown seaweeds. So all of these are based on external morphology for their identification. And now the red seaweeds. So the species distribution, the total macroalga taxa, which is 62 species, represented 6% of the 1,065 reported taxa from the Philippines. So this was the updated checklist of Lastimosa and Santianes. So this is the recent study. So 1,065, so 6%. And of these 62 species, 46.77% constitute the green algae, 29% for the brown algae, and 24.19% for the red algae. So these are the 9 species that are commonly found in all stations. And this can be due to the fact that probably they're macroalgal, based on their macroalgal spores. So species diversity and abundance of macroalgae. So this is the diversity profile in 8 sampling stations in Siargao Island, Surigao del Norte. So the diversity indices were the taxa, or the number of species for each sampling site, a total of 8 sampling sites, the individuals, the dominance index, the diversity index, and the evenness index. So as you can see here, the evenness index and dominance index are high, meaning that there is no dominant species in the area, and the diversity, since the evenness index is considered when there is greater than 0.6, so it is considered low evenness. And now the diversity index, which is also known as the Shannon-Wiener, all of the values in the 8 sampling stations are higher than 2.0, meaning there is a high diversity. So in here, the species diversity and abundance of macroalgae. So the Katangnan and the Poblacion 1, which is the CAT and the POB1, has the highest green macroalgae in terms of the number or total individuals, followed by the brown algae found in Malinaw or the MAL, and the Caridad or the CAR, and lastly, the red algae, which has the highest contribution of individuals in Tigasao or the TIG, and the Pacifico or PAC. So here, the species diversity and abundance of macroalgae. So these are the three highest three highest seaweeds that have a highest IVI or the importance value index, which IVI is used for the community structure. So the Porteria hornemani, the 66.43%, the Tricleocarba fragilis, and the Gelidiella acerosa. So in these 3 seaweeds, they are all red seaweeds, and their high IVI or importance value index can be due to the fact that they can be due to the growth rate, herbivore tolerance, reproduction, and successful attachment to a range of a variety of substrates. And as you all know, these 3 seaweeds are also very important or economically important because each of them have their own important functions. Firstly, the Porteria hornemani, which is identified to have antioxidant property, the Tricleocarpa fragilis, which was known to have an antibacterial property, and lastly, the Gelidiella recerosa was a good food source or known to have an analgesic, anti-cancer, and neuroprotective properties. So, the summary conclusion and recommendation results showed that there were 62 macroalgal species identified and classified in Siargao Island. The green algae were the richest group, followed by the brown, while the red was the least. Siargao had high to moderate diversity, and the evenness index was low, indicating that there were no dominating species found in the macroalgal population. So the top 3 species were Portera, Tricleocarpa, and fragilis, and the effects of season on the macroalgal population is also an interesting part that needs to be considered in the future research, and further ecological studies on the seaweed resources may provide useful data for the conservation of marine algal resources in this locality, and also for recommendation. It is also recommended to use molecular techniques to make the identification of macroalgae more faster and accurate. That is all. Thank you.
Thank you, Ticia. I think this is just a kind of preliminary work, right? The just first survey, first investigation, I feel that. What? Anyway, okay, please, I'd like to have some people, ecologists or algae. Okay, please.
We've heard the word sustainability over and over again. How sustainable do you think the local people could use the important brown algae? I ask this because brown algae soup, which I've had in Okinawa, is very healthy, keeps me awake all day and all night. Sustainable use of these brown algae. Could you comment on that? Thank you.
So as far as I know, that's still new to me about the brown seaweed soup, since brown seaweeds is known in the Philippines to be as a feed. It's used as a feed, like example for the animals like the pigs, they are used as a feed, the brown seaweeds. For sustainability, in the area, in Siargao Island specifically, the people there are really not yet educated or know about the importance of seaweeds. So probably the first step for us to do in that location is to educate them on what the economical and ecological importance of the seaweeds are.
Thank you, Ticia. Please, we'd like to thank Ticia for her presentation.
So may I invite the third speaker, Kun Titilat Mijan. Her topic of talk is morphological and genetic analysis of stingrays in the Tila-Trigon complex in Thai waters. Titilat, please.
Good afternoon, everyone. I would like to share the results. I would like to share the result on morphological and genetic analysis of Tila-Trigon stingray in Thai waters. Ray is important to marine ecosystems. Their abundance is declined rapidly. Thailand and other countries are taking actions, but biological information is still limited. The research focuses on the stingray in the Tila-Trigon complex in Thai waters because of its confusing taxonomy. Stingray and derivatives in the order Malebatiformis is the largest group of bacteria. It contains 11 families worldwide and 9 families in Thai waters. Stingray is the Yacidae family consists of 20 species worldwide and 45 species in the Thai waters. The more recent revisions, last SL 2016, resulted in the major reclassifications of the family with more genera and species being declined. A former classification of this genus is a single species of Dadiatis sugii. A recent classification inside this group of the distinct genus of Theratigons, which consists of at least 4 species worldwide. In Thai water, these are at least 3 species. Last SL 2016 proposed 2 additional species to Theratigons sugii, namely Theratigons cocci and Theratigons biasa. Theratigons sugii is common in the South China Sea and Thai waters. Theratigons biasa is present in southern Thailand, Gulf of Thailand, and Andaman Sea. And Theratigons cocci is only present in the Andaman Sea and the Indian Oceans. However, specimen included in this report does not represent the entire Thai water, and the diagnostic characteristics overlaps among these species. The objectives of this study are to clarify the taxonomy of Theratigons complex in Thai waters based on 42 morphological characteristics and DNA barcodes, and examine phylogenous relationships within this group based on nucleotides, sequence of 2 mitochondrial genes, CO1 and ND2. There are at least 7 diagnostic characters for this species in the existing taxonomy. This character covers this has snout shapes and lengths and level-length proportions. Through these shapes, tail length and skin fold shape and length. Reverse morphological and morphometric character and genetic variations. Samples were obtained from various sources, including research vessels, fish markets, and fishing harbors in the Gulf of Thailand and Andaman Sea. These specimens were identified to species levels. For morphological study, we examined 1 and 4 specimens of each putative species from Andaman Sea and 20 Theratigons biasa specimens from Gulf of Thailand. For genetic study, we obtained 24 CO1 sequences and 16 ND2 sequences from Andaman Sea and 31 CO1 sequences of 33 ND2 sequences from Gulf of Thailand. We measured 39 morphometric characteristics, including 18 haze characters, 10 disc shapes characters, 13 tail-related characters. All are reported as a proportions to these widths. We use PCA to evaluate the similarity of the characteristics among key protective species. Based on the morphology characters, Telatygon biasa tend to have shorter snout and tail, but wider distinguished other two species, Telatygon cocili and Telatygon sugii, very similar. PCA suggested at led two groups. Group 1 consists of mostly Telatygon biasa, simple form of Thailand. Group 2 consists of 3 species. Most specimens from Andaman Sea. Overlapping part the two groups include specimens from Telatygon biasa, from Gulf of Thailand and Andaman Sea, and Telatygon sugii from Andaman Sea. Fin clips were collected and preserved in absolution ethanol. DNA sample were extracted using salt extractions, DNA barcodes based on CO1 and ND2s were amplified using PCR. PCR products were checked by electrophoresis and purified and then submitted to sequencing. All sequences were analyzed using mega version 11. We calculate genetic diversity of each gene for specimens from Thai water and those deposited in GenBank, the phylogenetic tree, were contractions using maximum likelihoods. We got more than 600 base pair. We got more than 600 base pair for CO1s and more than 900 base pair for ND2s. The variations was about 3% for CO1 and 4% for ND2, the Thai sample. Maximum likelihood phylogenesis analysis based on ND2 sequencing suggests 3 groups. Group 1 consists the single Andaman hypothesis from our study and Telatygon sugii from Andaman Sea in South China Sea. Group 2 consists of hypothesis from Gulf of Thailand and Telatygon biasa from South China Sea. Group 3 consists of the GenBank sequencing of Telatygon sugii and South China Sea. Similarly, the maximum likelihood phylogenetic tree based on CO1 contains 3 groups. Group 1 consists the haplotype from Gulf of Thailand. Consists of the Andaman haplotype with some GenBank sequencing of Telatygon sugii and Telatygon biasa from Malaysia. Group 2 consists of the haplotype from the Gulf of Thailand with some GenBank sequencing of Telatygon sugii. Group 3 consists of the GenBank sequencing of Telatygon from the South China Sea. Genetic distance between in the Andaman and Gulf of Thailand haplotypes were 0.005 for CO1 and 0.015 for ND2. Genetic distance between Andaman haplotypes and Tela-Taigon Suji in South China Sea were 0.014 for CO1, 0.029 for ND2. Morphological data and genetic data support the clearly identification Tela-Taigon bears from Gulf of Thailand. This group differs from the Andaman groups. We cannot conclude the presence of Tela-Taigon bears in the Andaman Sea because don't have genetic data. The remaining two species in the Andaman Sea were not referenced based on morphological and genetic data. It is possible that our sample from the Andaman Sea is actually only one species. Morphology and genetics of Tela-Taigon bears in Gulf of Thailand is different from Tela-Taigon coastally in the Andaman Sea. Specimens from the Gulf of Thailand are likely Tela-Taigon bears, but samples for the Andaman Sea we still not sure. Kajang-Dara et al. 2022 reports Tela-Taigon bears only the southern Thailand, but our research confirm the distribution of Tela-Taigon bears extend to the upper Gulf of Thailand. Due to the lack of sample for DNA analysis, we cannot confirm the presence of Tela-Taigon bears in the Andaman Sea. Tela-Taigon Suji and Tela-Taigon coastally from the Andaman Sea cannot clearly form the another based on the genetic and morphologies. This is possible that Andaman groups only consist of Tela-Taigon Suji coastally and samples are genetically similar and very distinct from Tela-Taigon Suji in the South China Sea. To verify if these species co-occur in the Andaman Sea and Tela-Taigon occur in the Gulf of Thailand, we need additional specimens representing all morpho species in Thai waters. Inconstant naming of the gen bank sequences creates some confusion or some sequence to be submitted should evaluate and revise on the regular basis. I would like to thank the National Research Council in the technical support. Thank you.
Probably one short patience is allowed. Any patient? So please.
ขอบคุณครับ Thank you very much for the Stingray talk. It looks as if you've got fresh, new, powerful evidence that these species aren't genetically isolated. They are interbreeding even on both sides of the Thai peninsula. Looks like your work has indicated that we're not sure that they're separate species. Thank you.
Tela-Taigon bears Gulf of Thailand, from Gulf of Thailand, and Andaman Sea, not sure.
Thank you. I'm not sure. Couldn't properly hear. Thank you.
Thank you.
So thank you for our speaker join me. Yes.
Okay, come to the next papers from Dalika Kongrit, characterization of the potential Amicola topis, novel species 505 and CM21R, isolated from pharmaceutical plant roots, Cysampilos, Palaita, and Cariota mitis. Please welcome Dalika Kongrit.
Good afternoon, everyone. My name is Darika Khongrit, a lecturer and researcher at Prince of Songkhla University, Surat Thani campus. Today, I would like to present to you my research on the characterization of potentially novel Amicolatopsis species 505 and CM201R, isolated from velvet leaf and fishtail palm. There is an ongoing need for novel biologically active compounds capable of combating drug-resistant pathogens and demonstrating efficacy against tumor and cancer cells. This research specifically focuses on exploring new derivatives within the enzymycin antibiotic group. A key aspect of the investigation involves the isolation of actinomyces, a filamentous bacteria. Isolate 505 and CM201R as a key aspect of the investigation. The reason why we chose the pharmaceutical plants as a material in this study because they are relatively unexploited resources. Another reason is that the potential genetic exchange between pharmaceutical plants and endophytic actinomyces, yielding similar bioactive compounds to those of their hosts. Pharmaceutical plants are abundant sources. The utilization of pharmaceutical plant roots is grounded in their significance as a source of vital nutrients, including sugar, organic acids, and amino acids that may attract microbes, including actinomyces. In exchange, they protect the plant against pathogens by releasing antimicrobial compounds. This slide elaborates on the chemical structure of antibiotic within the enzymycin group. The highlighted ringed functional group in the image showcase specific characteristics of subcompounds, namely the benzoic, naphthalinic, and mitomycin C groups. In this study, rifampin will serve as a screening tool to isolate bacteria within the actinomyces group, possessing antibiotic-resistant capabilities. Classified as a naphthalinic enzymycin, rifampin is chosen for its antibiotic-resistant properties, making it instrumental in selecting new derivatives. The reason behind this choice lies in the likelihood that microorganisms resistant to antibiotics may possess the ability to produce antibiotics themselves as a cell protection mechanism. This table provides a comparison between the rifampin-resistant method and alternative approaches. Notably, screening based on rifampin resistance demonstrates the highest success rate in identifying enzymycin. The objectives of this study are as follows. One is to isolate the potential novel enzymycin derivative producers using rifampin-resistant mediated isolation from actinomyces. Two, is to identify enzymycin derivative producer and classify types of enzymycin derivative from the acquired rifampin resistant actinomyces using AHBA synthase or DIFK gene. The third is to classify species in the putative enzymycin derivative producers using polyphasic taxonomy approach. The key points regarding screening based on rifampin resistance are as follows. Rifampin resistant based screening. Second is to use PCR to screen DIFK gene. And the third one is preliminary staging of antimicrobial test properties using a diffusion assay and LC-MS analysis. Rifampin at a concentration of 20 mg per liter was used to isolate endophytic actinomyces strains resistant to the antibiotic. This figure features identification of DIFK and classification of enzymycin derivative based on phylogenetic relevance of DIFK. Preparation of crude extracts from isolate 505 and CM201R and diffusion analysis of antimicrobial properties was shown in figure A and B, respectively. Details of genotypic and phenotypic characterizations are described in this slide. The slide shows the identification. This slide shows the identification of 16 RNA genes from isolate 505 and CM201R, followed by phylogenetic tree construction. This slide showcases analyzing procedure of ANI, average nucleotide identity, and digital DNA hybridization values, as well as secondary metabolite gene cluster BGCS of 505 and CM201R from genomic data. This slide shows the detailed characterization of the morphology, physiology, and biochemistry of strain 505 and CM201R. Analysis procedures of wholesale sugar by thin layer chromatography. Analyzing procedures of diaminophenolic acid, wholesale fatty acid pattern by gas chromatography analysis. Results. 3 out of 51 isolates were found to contain DIFK, indicating enzymycin biosynthetic capability. Upon analyzing their sequence, both enzymycin produced from 505 and CM201R were observed to belong to the naphthalinic group. Both 505 and CM201R were shown to have antimicrobial activities against tested pathogens. LC-MS analysis of a crude extract prepared from CM201R showed the presence of rifampin. 16 RNA genes from 505 and CM201R were found to be closely related to Hemicholatosis plumensis SBHS STRP1 type strain. ANI values from both 505 and CM201R indicated that both strains could be classified as novel species, as the values are lower than the 55 to 96% threshold. Similarly, digital DNA hybridization values for 505 and CM201R also suggested the possibility of new species classification, given that these values are below the 70% threshold. As we could not identify enzymycin derivative from 505 using LC-MS analysis, Judging from BGC biosynthetic gene cluster predictions, 505 contains 60% biosynthetic gene cluster similar to rifamorpholine, which belongs to the naphthalinic enzymycin class. It is possible that 505 may produce similar kinds of compounds. This slide features the characteristic hyphae morphology of strain 505 and CM201R. Both 505 and CM201R contain mesodiaminopimelic acid, a key component of bacterial cell wall in the genus Hemicolatopsis. Both 505 and CM201R show typical sugars, polar lipids, and fatty acid profiles in the genus Hemicolatopsis. These slides provide a summary of morphology, physiology, biochemistry, chemotaxonomy studies of 505 and CM201R, comparing them with the Hemicolatopsis plumensis type strain. In conclusion, antibiotic-resistant mediated isolation has proved to be efficient in the discovery of enzymycin from the chosen pharmaceutical plants. Analyzing RIFK and biosynthetic genes cluster BGCS help identifying the enzymycin derivatives synthesized in CM201R. From phenotypic and genotypic studies, including 16S rRNA phylogenetic tree construction, analyzing genetic parameters such as NI and digital DNA hybridization values, reveal the two newly identified isolate 505 and CM201R as belong to a novel within the genus Hemicolatopsis, a rare member of Actinomyces. Thank you very much for your attention.
Thank you, Dalika. We have time for 2 questions or comments. Please.
May I start?
Yes,
one question.
Yes.
One simple question. So it seems that at first you want to get the new producer for new antibiotics.
Yes, that's right.
Are these two strains produce new derivatives of antibiotics?
The first one, CM201R, seems to produce the classical antibiotic, but for 505, still we are not know. We have to prove further because it seems to produce a new derivative of enzymycin antibiotic.
Any more comments or questions?
If more, I have
more
questions.
Please.
For proposal of your new species of Actinomyces, what you are going to do more? I mean, the whole genome analysis or something like that?
We did whole genome analysis, especially the genome sequence, but we need more data. For example, the menaquinone is a kind of vitamin K2, which exists in cell wall in bacteria. So this is a key parameter to use as a group this strain as the genus Hemicolatopsis. So we need more experiment.
Thank you, Dr. Dalika Konglit. Yeah, congratulations. I just heard that you just got PhD past the diploma.
So our last speaker is Dr. Adnarong Mano Suthirit. He's from Burapha University. His talk will be about the results of development of learning curriculum on biodiversity and cultural heritage of Eastern Thailand. Please, Dr.
สวัสดีครับ This research is, I'm not sure about in the size because I'm from Faculty of Education, but we can join, we can collab. Okay. This research project is we have too many contents and too many data from many faculties in the Burapha University, such as Faculty of Science, Faculty of Allied Science, maybe Marine Technology, and like this. I will show about the agenda of this presentation. First of all, I want to introduce about the plant genetic conservation project under the Royal Institution of Her Royal Highness Princess Mahachakri Sirinthorn ISPG, and we call in Thai OPSOTO project. Burapha University is located in the eastern region of Thailand. There are too many researchers to serve in the ISPG project, and this project in a big project, there are too many researchers in this project to operate on this project plan. In this project, this is a second period of operating, because in the last year we document research and we need analysis to stakeholder in the eastern region. This is our second year of operate to create the curriculum, learning curriculum about diversity and heritage of eastern region. And objective number 2, to evaluate lesson plan, because this curriculum to use in the school for teachers and students in school. And the last objective is to evaluate the elements of curriculum. Research scope area. After the period 1 from the last year, this is period 2. We have got the prototype of curriculum. And in the curriculum, I include learning management plan, or we call in the lesson plan for the teacher, and the elements of curriculum in this curriculum. On the target group of this research, when finish the lesson plan and when finish the curriculum, we evaluate by the expert in the 3 experts in the 2 curriculums in this curriculum. Because the first curriculum is about the diversity of natural resources and is from the Chanthaburi campus, is about the Eastern coastal ecosystem for the secondary school student. This is the assessment topic. This slide is show about evaluate results. Overall was good. And the next for the lesson plan of the collecting and classify specimen of mollusks in the eastern region for the secondary school student is work good. And this one is a result of learning curriculum competence. Overall is good. And this slide show the result of assessment topic of curriculum of collecting and classify specimen of mollusks in the eastern region for secondary school students. When we finish about the evaluate process, we have two formats of implement of the curriculum. The first one is format in the boot camp in two days, 1 night at the campus of Burapha University in Bang San and Chanthaburi province. And the format 2 is we put on the data information about clip video, pre-test and post-test, or the content, everything about curriculum on the e-learning platform. This is information about the first curriculum. We have 8 sub-learning units. It's about ecosystem in the eastern area. And the next curriculum is, there are 5 sub-learning units. It's about mollusks. Okay. And all of evaluation, it was good. And now we ready to implementation in the next step, because now in the edit and improve the curriculum on the boot camp and e-learning platform. You can see in the picture, this year and period 2 is produced about the clip video and to go to the field study to create the clip video in the eastern area. This is a suggestion for the research. There should be research and development of media that is generated from curriculum and the result of using the curriculum for learning regarding the diversity of natural resource and cultural heritage of the eastern region that is consistent with the condition of technological change afterwards. Curriculum evaluation study should be conducted during the curriculum implement process in order to leading to know the curriculum quality or efficiency of the course with the intention of using the information as guideline for develop the curriculum. Okay, I've finished my presentation. It's too fast. Yeah.
So suggestion. Yes.
This is a second step of to produce, to develop the curriculum.
Any questions?
I have one curious, that why did you select the mollusk specimen? Classification on your result. What?
In the first period of the research project, we have needs analysis by the research of Burapha University. There are many fields of researcher in science, in everything about marine science. Some research about bees, about ants, about crab, like this.
Yeah, I know that, but how significant you have select?
I survey.
Mollusk.
Sir, yes.
How significant?
I choose the pick up in the first 1 to in the 3 of first selected from the school and the stakeholder. Before we create the curriculum, we need survey by Google form. Send the survey form to the student, to the school in the eastern region, and info back to the research team, and we have information about interested of topic like this.
Thank you very much.
ครับผม
Any more patients? So if not, join me to thank Dr. Narong. Thank you very much.
Thank you.
Okay, ladies and gentlemen, to wrap up this session, I want to express my heartfelt gratitude to our speakers. And your insights have been invaluable, expanding our understanding of the intricate relationship between the biodiversity and ecosystem conservation and services. And a special thanks to Professor Dr. Savitri Limthong and Professor Dr. Somsak Panha, our session chair and co-chair, for guiding us through the wealth of knowledge presented today. Thank you. Please give a big applause to Professor. Okay, and to the audience, thank you very much for your being active in this conference. And as we close this session, I encourage you to explore the poster presentation session located on the ground floor of the conference hall. The topics cover this conference, the biodiversity-related themes, including ecosystem conservation and services, climate change, and BCG economy models. And please feel free to take the exits behind this stage. You can turn right or left and walk along the way for the poster area. And following this, we have a welcome reception in front of the hall at around 6:15. It's an opportunity to network and share insights in a more relaxed atmosphere. And please enjoy buffet-style dinner and special dishes, pork and beef satay and a special one from Titlada Vocational School cuisine, boat noodles, and also other things that you can enjoy, including special music treats from Kasetsart University. Okay, and we finished day one of the International Conference of Biodiversity 2023. Your presence had made this day truly special, and I look forward to seeing you again tomorrow. Thank you, and see you at the welcome reception. May I invite all speakers for this session and oral presentation with the chair and co-chair to have a group picture on the stage, please.
you.